force going in both directions. What's this spot here called?
The Ori.
Alright, so start off in the Ori. So what's the first thing that's gonna happen?
So wouldn't it be a Primace first?
Primace first. Okay, so Primace is gonna lay a primer here, and lay a primer here.
Okay.
Okay, so what end is this?
So that would be the yeah, 5 prime, right? Or no?
3.
Why is this the 3 and this 5?
Oh, because like it has to be like opposite of this.
Yeah, right, so anytime you have 2 strands,
Right.
remember they're always integral. Okay, so what's this one?
5,
This one?
3.
Okay. So, right, well, your DNA polymerase 3.
Okay.
How are you gonna build right now from those 2 primers?
Okay, so DNA poly always has to start with a OH group from the 3 prime, right? So wouldn't it be like for example, like okay, since it's the leading strand, it goes all the way, right?
Okay, so you're gonna, remember you're building off the primer, so this should be
Yeah.
connected.
All connected.
Yeah, because it's all one piece now. Actually, I have this weird hybrid of DNA and
Nice.
RNA together. Alright.
Okay. And same thing with this one since it has an OH group right here.
Mhm.
So,
Okay. So, now we have these lagging strands, right? Okay, who wants to be primates? See ya.
Oh, Melanie.
Who wants to be primates?
I'll-
Let's switch it up. Sure, how about one more?
Here?
Sure. Alright. And? Got one other.
Oh, okay.
Alright. DNA polymerase 1, do your thing.
It's for poly 1?
Yep.
Okay, so this one's
So draw an arrowhead at the end of your, like draw an arrowhead here, just so you.
Like that?
Yeah.
Okay.
Just easier to keep track of.
Okay. So this would be a 5 prime, right? They're not connected, right?
They are connected.
Oh, they are?
Yeah, so remember, it has to build off the 3' end. So it's literally covalently linking onto 3' end.
Because I thought it wasn't really like initially connected because it needs the base pairs or it's connected for the poly 1 or no?
It has to build off of there. Otherwise it couldn't build. But here it's not connected.
Oh, it's big.
So that part's the one that's not connected.
Right.
Oh, okay.
So add an arrowhead on there. Whatever you have to do.
3 prime, right?
Yep. Add your
Professor,
arrowhead. Yeah.
I thought the Ori like began in the beginning, like where the 5' is, or could it start?
The origin is where, so remember we had our double-stranded DNA like this? And I said if this is the origin, this is where we're gonna make a bubble. Right, and then that bubble is just gonna get bigger.
The worksheet we were doing was like the 1/2 of this. You know what I'm saying?
Right, so you've just been looking at one replication fork,
Oh, okay.
which is half. Alright, so add an arrowhead on there and on there. Okay, and then do the same thing for those guys. There we go. Okay, so we have 2 problems. The first is I have RNA when I'm not supposed to have RNA. The second is I have the lagging strands in pieces. Okay, so DNA ligase can't do anything yet. Who do we need first?
Poly 1?
Poly 1. Okay, so who wants to be poly 1? Somebody who hasn't volunteered yet. Eeny, meeny, miny.
Come on. We went over this yesterday, like.
I don't like it.
I'll do it.
Alright, go for it.
Okay, so poly 1. So we would have to. Is it blue?
So poly 1 just goes back to replace the pairs, so it could be be like solved. Never mind. I'm wrong.
So you're on the right track. We have to remove the RNA. So remember, poly 1 can chew and lay down in the same direction. Right, so if this is my poly 1, right, I'm going to be chewing this direction and building this strand at the same time. Go for it.
Okay, so poly 1 is basically just change the RNA prime or primer and then replace it with nucleotides, right? That's the purpose of it?
Replace with which kind of nucleotides?
Like the base pairs that are similar to this and this.
Right,
Right.
but that's what was already there. There are nucleotides that are complementary to this. So what's the difference between the nucleotides that are already there and the one that DNA polymerase
Isn't this
is
because it's
gonna
RNA?
do? Exactly.
Oh, okay. So let me change
Now, we can't do it here yet in this diagram.
that. Okay.
So, put our primer back on there. You know, I'll show you why in a second.
Mm-hmm.
You kind of already know why, but we'll. So
Okay.
where can I do it?
To change?
Why can't I do it here? DNA polymerase 1 can't work here yet.
Oh, doesn't it go from 3
to
no, no,
5.
was it 3 to 5 or 5? Wait.
3 to 5.
Don't tell me. 3 to
Yep.
5?
They always go 5 to 3.
5 to 3.
Everything is built 5 to 3. But what do I have to build onto? I can't build from scratch. I always have to build onto.
Oh, wait, don't tell me. Crap, I forgot. An OH group? No?
Yeah, you always have to have a 3' end.
Okay.
Is there something hanging off here to build onto?
No.
No. So right now we couldn't do it here, but is there something here?
Yeah?
Yeah, there's a 3' end there.
Oh.
Right? So I can build off of that, and as I build this direction, I'm going to chew this up. Right? But as I'm chewing that up, what else is going on?
So now
Right.
now we're connected? Well, technically not connected because it
It's
needs
not-
ligase,
yeah,
right?
so draw
Yeah.
an arrowhead.
Okay.
Right, so we've solved one problem. Right, remember we had 2 problems. One is we have RNA when we're not supposed to have it, and 2, I have all these pieces. So the first thing we did is we replaced the RNA. So we can do that here, right? I have a 3' OH, so I can build off of this and keep going and replace the primer.
So like that?
Mm-mm.
Like connected?
Where should it be connected? Where should it be connected and where shouldn't it be connected?
Oh, okay. Since it starts here. Oh, wait, no. Yeah, it starts at a 5 prime. Here. So like this or no?
Mm-mm. Let's put the primer back. Right, so here's my primer.
Mm-hmm.
This is the 5' end of my primer. This is the 3' end of my piece here.
Because if it goes from 5 prime to 3 prime, wouldn't it be like...
When you say goes, what do you mean goes?
So wouldn't it start from right here and it's just this is the 3 prime technically? The arrow points that way? Is that what you mean?
We're always building 5' to 3'.
Mm-hmm.
So where's the 3' hydroxyl that you're going to add on to?
It's gonna be right here, isn't it? Because this is the 3 prime.
So what's hanging off the end here?
So that's a OH group, right?
Uh-huh.
So this would be like the what was it? The phosphate group? Was it? Right?
Right, but you always add on to which one?
To the OH group.
You always add on to the OH
Uh-huh.
group. So which one of those are you physically extending?
I don't know what you mean by physically extending.
So here's our base, sugar. Right, here's my phosphate, here's my OH. Right, if I'm gonna add another nucleotide on here, right,
Mhm.
this is my 3' OH.
Right.
So I'm always building this bond.
Mhm.
So I physically added on to the 3' end, I'm extending my chain this direction.
Oh, okay.
So if my 3' OH is hanging off right here,
So you're saying it's gonna be like this?
bingo.
Okay.
Good.
Oh, okay. Okay.
Right, because you're always attaching on to the 3' end. So you're extending off the 3' end.
But since there's no more RNA or primer over here, wouldn't this just connect, or no?
It can't, right? Remember, can only add, if we're adding onto the 3' OH, we're always adding over here,
Right.
right? I can't add onto a 5' end, right? Because when I get to here, right, when I get to this point, it just added this to another 3' OH.
Mhm.
What's over here? Phosphate. It can't add on to the phosphate. It has to bring in a nucleotide to add on this way.
And that's what the ligase is for, or the poly?
Exactly.
Okay.
So polymerase can't do that if there's something already here. Only ligase can do that.
Okay. But what about the 5' up there?
Okay, so right now we can stitch this together.
Mm-hmm.
Right, so do the same thing for those. Okay, right, and if we took this all the way out to the end, you see there's a whole other problem that we have. That's another conversation. Right, so where is my leading strand and where is my lagging strand?
Okay.
Wouldn't this be the leading strand?
Yeah, so we got leading here and leading here, so I got lagging
Lagging
here
here.
and lagging here.
Uh-huh.
So ligase is gonna do what?
Would you like connect these?
Yeah, so now we're gonna connect everything.
Okay.
But the only reason that I connect this lagging to this piece of leading is because I took that primer that was here and used DNA Pol 1 to get rid of it. Same thing here.
I'm sorry, can you repeat that one more time?
Right, so for this, if I just looked at this fork, I had a leading strand and a lagging strand. And if I just looked at this fork, I had a lagging strand and a leading strand. But I connect all of that the same way I connected the laggings together, right? DNA Pol 1 has to get rid of the primer.
So you're saying poly 1, all it does is like connecting the leading and lagging strand from here?
Isn't poly one?
It's connecting everything.
Okay, but for the purpose of the leading strand, that's all it does for the leading strand, right?
Correct.
Okay, but for the lagging strand, of course, it like connects like.
Yeah, you actually have
Okay.
to use it to build your lagging strand.
Okay, okay.
Yes.
Okay. So, what about these though?
Ah, okay, so what happens? Okay, so let's follow this logic through. I started here with the primer and here with the primer. Right, so then we built this one continuously. And then we had primers all along here, so I'll just do this. So primers all on here. Let's say like this. Right, so this was the leading strand here. So we did this. Right, and then here we had to do this. Okay, so DNA polymerase 1 we said is going to, and then ligate it together, ligase, right? Remove the primer, extend, ligate together. This is just the exact same thing that we just did a minute ago. You see complication here?
No.
What am I gonna do with this? Well, DNA polymerase 1 will go in and chew it up. But what can't it do?
Glue it to put together.
What do you mean?
Like the ligase. Like that.
So this is already all stitched together here. But what are we supposed to do?
Oh, is that the part? Because I was reading some part like it's always gonna be shorter than the actual strand, or something like that.
Yeah,
It's like
right.
relatively shorter.
So remember when we filled this in, right? I had a 3' OH here,
Mhm.
and I could build off of that. When I destroyed the primer, I could build off of this to extend it. Is there anything over here to extend off of? There's nothing there. So when we chew up that primer, this new strand is a little bit shorter on this side, and this new strand is a little bit shorter on that side. And if we were to replicate this again, right,
It gets
the ends
shorter
would
and
get
shorter,
shorter again. So we have a problem. How do we solve that problem?
I just read this yesterday.
You remember what we call the ends of our chromosomes? What that region's called?
Telomeres. Telomere?
So telomere is the region. So what's the enzyme that fixes this problem?
Telomeres.
Telomerase. Right, so telomerase can fill that in when DNA polymerase can't. And we didn't talk about how, and I don't expect you to know how. But telomerase is what can fill that in. So, if we didn't have telomerase expressed, so if my cell doesn't turn the telomerase gene on, what's going to happen to my DNA over time?
It's gonna get shorter.
It's going to get shorter and shorter and shorter. Actually, in most cells, that's exactly what happens. Most cells in your body does not have telomerase on, which is one of the reasons why your cells age. But, you know, when you're like one cell, and then you divide into 2 cells and 4 cells, you have to do a lot of cell division. So early in development, your telomerase genes are all turned on, and you're making sure you protect the ends of your DNA. But as you get older, this gene gets shut off, and so you don't do that anymore. Can you imagine a cell that would want to turn that gene on so that it could just divide over and over and over again? What kind of cell might that be?
Stem cell?
Stem cell. That's right, so you have stem cells, and stem cells also express this because they have to divide all the time. Can you think of another cell type that divides a lot?
Cancer cell?
Yeah, cancer cells. Cancer cells are normal cells that figure out a way to turn that on. They're not supposed to have it
Wow,
on. Yeah.
that's
They're
crazy.
tricky.
So that's why, like, sometimes when people get chemotherapy, they get it back again, like, sometimes, like,
I don't know if
when they're
they actually do that.
cancer-free and then, like, a few years later, they have it again.
Usually what happens in that case is you didn't get rid of everything. So you might have
Because
killed...
of that, because of the telomerase?
Usually it's not because of telomerase. So in order for it to become a cancer cell where it can divide uncontrollably, it already has this. So all of this, or most of the cancer cells will already have this expressed. But what happens is, it's kind of like when you take antibiotics, you might kill 99% of stuff, but not 100%. And all it takes is the ones that live to keep going. So usually when you get a relapse, the reason why is because there are some cells that survive the chemo, whatever treatment it was. So it might take a long time for them to grow back, right? Because you killed a lot of them, but you just haven't killed everything typically. Yeah, but one of the ways that they're able to become a cancer cell oftentimes is because they are turning that gene on when they're not supposed to. Very few cells in your body should be expressing that gene in adulthood.
I also want to ask about exonucleus activity.
Mm-hmm.
So what was like because I remember reading something like 5 to 3, exonucleus activity 3 to 5,
Mm-hmm.
and then endonucleus, I'm kind of like
Okay.
iffy about it.
So here's our primer. And here's DNA polymerase 1 that needs to extend this. So what end is this here?
Pretty cool.
Okay. And this end?
5 prime.
5. Okay. So, if we're going to chew this up this direction as we build this direction, what activity is that? I'm chewing from the end. So is it endo or exo?
Isn't it endo because it's
Endo.
chewing?
Both endo and exo are chewing.
Oh, okay.
It's destroying the DNA, but it's
Exo?
chewing from the Y exo.
Because it's going that direction.
Well, it could be this way. It's chewing from the end.
Oh,
Right?
okay.
So what's an endonuclease? An endonuclease is like a restriction enzyme, right? So remember, a restriction enzyme is if I had like GAATTC, right? My restriction enzyme would cut the backbone here and here, right? It's like in the middle. It's not, it doesn't have to do anything from the end. This thing chews up right? Like it takes one nucleotide off at a time. It just keeps chewing it up, right? This thing just goes in and it makes a cut here and here. It's not cutting from the ends. So cutting from the end is exonuclease versus endo.
And which was by poly 1 or poly 3?
Okay, so which direction are we going here? If we're going this way in our diagram.
This go from 3 prime to 5 prime?
What end is this?
5 prime?
So which direction are we going?
To 5 prime?
This is the 5 prime. So
Oh.
we're going
The
to
third.
the,
Oh, to 3 prime.
so we're going from 5
2, 3.
to 3. So DNA polymerase, which DNA is this? Which polymerase? Right now?
That would be poly. The one eating the RNA?
Mm-hmm.
And poly 1?
So DNA polymerase 1 has to have which kind of exonuclease activity?
5 to 3?
5 to 3. Okay, but if you're DNA polymerase 1, and you're just going along and you put in a, I don't know, right, and you put in a T, and you're like, okay, that's good, and then you put in an A, and you're like, okay, that's good, and you put in a T, and you're like, oh snap, that's wrong, right? What end is this sticking off of here? We're going this way.
3 prime?
3 prime, so this is OH, right? And you realize this is not correct pairing. So I'm gonna chop this out. So I'm gonna go this way, and then I can go back to
Oh,
building this
proofread,
direction.
right?
That's proofreading.
Okay.
So if we're building this direction, which direction are we building?
5
5 to 3. We can only build 5 to 3. But for proofreading, I had to chop out going the opposite direction, which is
3 to 5.
3 to 5. Right, so DNA polymerase 1 chews and builds in the same direction. DNA polymerase 3 builds this way and then
And fixes backwards.
backwards.
What about this? Would this be poly 1 or poly 3?
Neither. That's
Oh.
like our restriction enzymes, the stuff we've been doing in lab,
Uh-huh.
like the EcoR1 and HindU3 and BAMH1 and all that stuff. Those are restriction enzymes, so they cut in a particular sequence. These guys don't care about the sequence, they just.
What about the sliding clamp? Doesn't it hold it together?
Yeah, so sliding clamp is gonna help to hold DNA polymerase on this thing, right? So you've got this thing that looks like a donut, sort of surrounding the DNA. I heard Dr. Brown showed you guys a decent animation in lab. I'm gonna steal this animation. This one is cool. Right, so, and then, well, there we go. Aha. The older I get, the better my drawings get. Right, so it's kind of like a ring around the DNA.
Does the sliding clamp have to be like in a specific place, or can it be like where it just holds?
What do you mean by a specific place? It's attached to DNA polymerase, right? So this is the clamp. Oftentimes they also call it the beta clamp. And this is DNA, all 3, for example, right? That whole thing is moving together. Because this guy is helping DNA polymerase stay on the DNA.
So it also goes from 5 to 3?
Yes, because it's moving with
Yeah.
DNA polymerase as it goes, yeah.
Mm-hmm.
So it's kind of along for the ride, if you want to think of it that way, but it's holding DNA polymerase to the DNA so it doesn't just like fall off too easily.
What else did we have? Is that it?
I think that was it, yeah.
I had something in my mind a while ago. What was it? Oh, so for the ligase part, for these parts right here, how does ligase specifically connect them?
All it's doing is, so let's imagine what's hanging off of here in the center.
3 prime?
So what's here? What functional group?
OH.
Okay, and what's hanging off of this end?
That'd be a phosphate.
Phosphate. Ready? Done. What kind of bond is it making?
Phosphodiester?
Yeah, it's just making the
Oh.
phosphodiester bond.
Oh wow, okay, I'm stupid.
Right,
Oh,
'cause
okay.
remember we had the two, so in the diagram we drew a few minutes ago, we had this.
Mhm.
Right, like that. the other way to think of it is, you were asking why can't we just build this?
Mm-hmm.
Another reason, typically, well, maybe I shouldn't complicate things. I'm not gonna complicate things. I'm being simple. Right. So, DNA ligase is gonna build that bond. So there's a phosphodiester bond. So it's making a phosphodiester bond just like DNA polymerase makes a phosphodiester bond, but it can do it at that gap where DNA polymerase can't. Because DNA polymerase can only build as it goes in one direction. And I think in the slides there's a diagram of that, like with the actual structures, that shows you like sort of this thing.
I think it says something like a phosphorylation, I think. Is that what it is?
We can take a look real quick if you want. I'll pull it up.
I think that was it. Was that it? Or-